All right, good morning, everybody, and welcome to our two-day symposium on mass spectrometry and epitranscriptomics and RNA biology. This is hosted by the NIH and the National Human Genome Research Institute Center for Genomic Information encoded by RNA modifications. I think everybody here knows that mass spectrometry has become an indispensable tool in RNA biology. And it's not just simply for getting a modification, fragmenting it, determining its identity, but also as a core analytical method for elucidating structures, dynamic regulatory functions in RNA sequences at nucleotide resolution. And this is particularly important for the progress of epitranscriptomics research. As everybody knows, there's over 170 known RNA modifications, and we are now systematically determining their chemical identities, their functions, their abundance, their turnovers, and each one of them can be very different. And so a lot of this is being enabled in large part through high resolution mass spec techniques. And unlike Illumina methods and other sequencing methods, mass spec can detect a broad range of modifications, and it doesn't rely on antibodies, doesn't rely on reverse transcriptases, and it provides quantitative base specific and chemically precise information. At least we hope, or we're trying to get that way. So these capabilities are really transforming how we approach problems in RNA modification. So this is why we're really excited about the symposium today. So we're going to begin today with a keynote lecture from Tom Suzuki. So Tom's a pioneer in RNA modification research. His work has defined many of the platforms and paradigms used across the field. So he's going to speak on tRNA modifications in health and disease, highlighting mechanistic insights, clinical relevance, and of course, methods. So in our right after our keynote, we're going to have our morning session, and this will include the speakers, which are Bi-Feng Yuan, who's going to talk about disease and environment-associated RNA modification patterns, Stefanie Kaiser, who will present novel methodologies that she's developing, Henning Urlaub, chemical cross-linking strategies for studying RNA modifications, and Yinsheng Wang. So he's going to address mechanistic roles of RNA methylation in repeat expansion disorders. After the lunch, we're going to have Scott Blanchard as the moderator, who's going to talk about mass spec-based RNA sequencing methods. We're going to hear from Kristen Kotmau, who's going to talk about recent developments in her lab. Shenglong Zhang, who's going to talk about methods for full-length RNA sequences by mass spectrometry. Benjamin Garcia, quantitative strategies for characterizing RNA modifications. And Chi Chen, who's going to talk about decoding these tRNA fragments. So we're going to conclude today with a panel. The panel is going to have many of the speakers. Kate Meyer is going to moderate this, and we're going to really address some of the key questions, like what do we need in the mass spec field? When is it the right time to use mass spec versus Illumina? What are the problems and limitations? And I think this will be sort of a open discussion. We're not going to push mass spec or push other methods. We're just going to try and see what's the best method at the best time. So I want to start today by introducing Tom Suzuki. And so I've had the pleasure of knowing Tom for many years, and so it's a great pleasure to introduce him. He's a professor of chemistry and biotechnology at the University of Tokyo. So he's internationally recognized for his contributions to RNA modification biology. Such key seminal work has come out of his lab. He has developed some of the most sensitive and robust mass spec-based techniques for analyzing modifications, both for global profiling and for single RNA interpretation. So he's discovered novel RNA modifications, which to me is always the most exciting thing, and their corresponding enzymes. He's described the first human diseases caused by tRNA, caused by RNA modifications, and he's continued to explore the molecular basis of mitochondrial dysfunction, tRNA, and translational control. Some of his recent papers, there was a landmark paper in Nature ChemBio recently about novel tRNA modifications in enhancing decoding fidelity. Paper I really liked about tRNA glycosylation in cell and its links to vertebrate growth. And if you want to learn more about the field, he has a great review in Nature Reviews Molecular Cell Biology a few years ago, which is really a foundational thing that we often give to students when they're starting research in this area. So with that, here's Tom, and he's going to speak on the expanding world of tRNA modifications in health and disease.
Thank you very much, Samie. for your kind introduction. Okay, so I'm gonna share my screen. Can you share? Can you see my screen?
Yeah, it's Tom, we can see it. And I
Okay.
forgot to tell everybody one important thing is that at the bottom of your screen, you should see a Q&A button. If you don't have the Q&A button, you can use the chat button, but you can answer, you can put your questions there. And at the end, we'll go through some of the questions, whatever we can get through.
Okay.
The speakers will all see a timer at the very end and then a ring when their talk is over.
Okay, thank you very much. So first of all, I'd like to thank Samie and other organizers for inviting me this exciting symposium. So I'm really honored to give an opening talk. Today I'm going to talk on our recent studies on tRNA modifications. So in the emerging field of RNA modification and epitranscriptomics, we have learned that RNA modification provide chemical diversity to simple RNA molecule, yielding a greater range of biological functions. Our tRNA modifications are well studied for more than half century. These modifications are required for proper functions of tRNA, in particular accurate decoding of genetic code and the stabilization of tRNA core structure. And the mRNA also have several modifications, including 5' cap structure, ion chains M6, and so on. And RNA modification confer physical chemical properties on RNA molecule and play a wide variety of biological functions as shown here. So we have now about 150 types of chemical modification found in various RNA molecules from all domains of life. So this is a distribution of RNA modification in 3 domains of life. So in this Venn diagram, a group of modification in the center are commonly found in archaea bacteria and eukaryote. As you see, there are a number of domain-specific RNA modifications. So diverse RNA modification are closely associated with evolution of life. So this slide shows you 16 RNA modifications discovered in this century. Modified groups are shown in red. As you can see, they have a wide variety of chemical modifications. So our group discovered reported 7 modification boxes here, including taurine containing modified uridine. And the lack of this modification causes human mitochondrial disease. And a couple of years ago, we discovered the 2' phosphouridine. So this is the first internal RNA phosphorylation found in zoophilic organisms. So very recently, we reported a novel cytidine modification named 2-aminobalaramidine, abatusin in short. So it was found in tRNA from plant organella and pathogenic bacteria. So I'm going to talk on this modification later in my talk. The chemical space of the RNA modification will continue to expand in the future. So here I want to tell you our platform technologies for RNA isolation and the direct analysis of RNA molecule by mass spectrometry. So this is our original method for RNA isolation, which we call reciprocal circulating chromatography, RCC in short. So this method enables us to isolate multiple individual RNA in parallel and automatically. For automated isolation of RNA, we assembled RCC machine, as shown here, which consists of liquid handling machine with custom-made head, design chip column, heater, agitator, and controller, which organizes all of the components. The DNA probe, complementary to the target RNA, is immobilized on this region, which is packed in this chip column. So in this device, we can set 8 chip columns in parallel. We put a total RNA solution in this vessel, and this machine automatically operates aspiration and pushing out and mixing. So these 3 steps are repeated number of times so that all total RNA solution circulates and pass through each column. And then target RNA is trapped in each column. After washing, the target RNA is eluted from the column separately. So as you can see, it works very well. We can automatically isolate tRNA, small non-coding RNA, and even microRNA. So to analyze RNA modification, we are devising capillary liquid chromatography, nanoelectrospray ionization mass spectrometry, which we call RNA mass spec. So they are suitable for analyzing a complex mixture of RNA fragments digested by our RNases. The RNA fragments are separated by this capillary column with flow rate of 300 nanoliter per minute. For high sensitive detection, we developed an online desalting system and optimized our solvent system and the LC setting and tuned our mass speedometer. So we can actually analyze sub-femtomole RNA molecule by this system. So this is an example of mass spec analysis of tRNA modification. We first analyze total nucleoside to know the species of modification contained in each tRNA. Then we analyze RNA fragments digested by RNase-T1 or RNase-A to detect modified fragments. And the position of each modification can be determined by collision-induced dissociation, CID in short. And then we map pseudo-origin biochemically using pseudo-origin specific chemical modification and primer extension followed by deep sequencing, because this is a mass silent modification. Alternatively, pseudo-origin can be derivatized by cyanidation or bisulfide method, and now it is analyzed by mass spec. And then finally, whole mass analysis of intact tRNA provide us useful information to validate our assignment. For mitochondrial tRNA, we isolated all 22 species from human placenta and the HeLa cells, and then we mapped all the modification in each tRNA by mass spec and the pseudo-origin sequencing. We identified 18 types of RNA modification at 137 positions in total species of mitochondrial tRNAs. And this is a integrated view of the mitochondrial tRNA modification. So many modifications can be found in the anticodon region. For cytoplasmic tRNA modification, it's a tough challenge for us because there are 429 tRNA genes encoded in the human genome. And there are many isodecoders whose sequences similar each other. And according to the deep sequencing of the human tRNAs, about half of the tRNA genes are actually transcribed in each cell. However, in the tRNA database, tRNA modifications have been mapped to only 18 species. So we started a project to isolate total species of cytoplasmic tRNA from human culture cell and perform comprehensive analysis of tRNA modifications. So we isolated about 180 species of cytoplasmic tRNAs from HEC293T cells by our RCC machine. And this is a summary of a cytoplasmic tRNA modification. And finally, we analyzed 179 cytoplasmic tRNA in total. We identified 45 types of RNA modification, including 4 novel modifications. And they are mapped to the 2,617 site in 179 species. So this number includes 456 pseudo-origin site. So 18.9% of the tRNA residues are modified, but these numbers are old. We are gonna revise them with our latest mapping data. So this is an integrated view of the human cytoplasmic tRNA modification. A wide variety of tRNA modification are clustered in the anticodon region, especially at the first position of the anticodon and position 37, which is next to the anticodon. These modifications play a critical role in accurate decoding and efficient translation. This is a very, very important reference data to study tRNA modification in various biological contexts by nanopore sequencing in the future. So here, let me explain our strategy to identify novel RNA modification. So we first isolate individual RNA from various sources and analyze RNA fragments digested by RNases by RNA mass spec to look for a unique molecular mass. So this is a clear indication of the presence of the novel RNA modification. Then we measured accurate mass of the nucleoside to determine its atomic composition. So this is very important information to determine the chemical structure. And then we probe the chemical structure of the nucleoside by collision-induced dissociation using mass spec. And we also perform metallic leveling experiment to determine the metallic substrate of each modification. And if we get a sufficient amount of the modified nucleoside, NMR measurement works very well to determine the chemical structure. Having this information together, we can narrow down the candidate structure And then we collaborate with nucleic acid chemists to synthesize the modified nucleoside. And finally, we determine the chemical structure by comparing the natural and the synthetic compound. So this is our typical strategy. So recently, we reported a novel cytidine modification named 2-aminovaleriamyridine, about 2C in short. It was found in anticodon of isoleucine tRNA responsible for AUA codon from plant mitochondria and chloroplast and the pathogenic bacteria, including Vibrio cholera and the Pseudomonas species. It has a long side chain with a terminal amido group. So the cryo-EM analysis tells us that this long side chain extends toward 3' direction of the messenger RNA and then makes a hydrogen bond with the mRNA residue, which is next to the AUA codon. So this is the molecular basis of AUA decoding mediated by ABA2C modification. So let me explain how to identify this modification. So this is a recipe to obtain a large quantity of the modified nucleoside for structure analysis. So I love an old-fashioned biochemistry. So we first bought a 40 kilogram of spinach from a supermarket near our campus. It's a huge amount of the material, but it's not so expensive. Then it was freeze-dried in a food company. Then we sent it to another company, food processing company, to grind it into powder. It's like a matcha powder. During this experiment, the entire lab was tinted green. Lab coats, wares, and sinks were green, and even my students are green. So it was very, very funny experience. Then total RNA was extracted from this powder, followed by DEA chromatography to obtain the small RNA fraction. Then we isolate the target tRNA by RCC, and finally we obtain sufficient amount of modified nucleoside for structure analysis, including mass spec, NMR, and the chemical synthesis of the authentic nucleoside. Then we determine ABA2C. So this is a procedure to identify the novel RNA modification in my laboratory. So in bacteria and archaea, AUA codon is led by tRNA having a cytidine modification at the first position of the anticodon as shown here. Almost all bacteria use a lysine modification. Lysine is a lysine conjugated cytidine derivative, while archaea have agmatogene, agma2C in short, which was also discovered in my laboratory many years ago. Now we have ABA2C. This is the third example of the modified cytidine found in plant organella and pathogenic bacteria. So these cytidine modifications commonly have long side chain with polar termini, which is attached to the C2 carbon of the cytosine base. To study a molecular basis of AUA decoding mediated by this cytidine modification, we solved the cryo-EM structure of the tRNA isoleucine, recognizing the AUA codon on the ribosome. So when we look at the decoding center at the S site, we can clearly see the codon-anticodon interaction and the cytidine modification. The resolution was good enough to model the tRNA modification. So all three modifications pair with a third adenine base of the codon via a unique CA geometry with a single hydrogen bond. So we clearly see the density of the long side chain, so and the which extend toward the 3' direction of the messenger RNA. And we can also explain the reason why these cytidine modifications avoid misdecoding of AUG codon due to straight crash with the long side chain. So the long side chain extend toward 3' direction of the messenger RNA in all three cases, and the polar termini makes a hydrogen bond with the 2' OH group of the mRNA residue, which is next to the AUA codon. So in the case of AGM2C, the polar termini guanidino residue makes an additional hydrogen bond with the ribosomal RNA residues. So the side chain of AGM2C is tightly fixed to this region. And also the density of this group is very, very strong. In terms of the straight complementarity, the long side chain nicely fit with the cleft between the mRNA residue and the ribosomal RNA residue. So the van der Waals contact and the hydrophobic interaction likely contribute to the efficient decoding of AUA codon. So based on this structural information, we here propose a chain and anchor model to facilitate AUA decoding mediated by the side modification. In this model, the long side chain looks like a chain, and the polar termini is an anchor. So this anchoring interaction of the side chain support the weak CA base pair with a single hydrogen bond. To study whether this additional hydrogen bond actually contribute to the AUA decoding, we introduced atomic mutation to the 2' OH group of the mRNA residue with 2' deoxy substitution, 2' O-methyl substitution, or fluor substitution. Then we performed SITRNA binding experiment with these modified messenger RNAs. As expected, 2' deoxy substitution reduced the tRNA binding ability due to loss of the hydrogen bond in both lysine and the ABA2C. And the 2' O-methyl group also reduced the binding ability because of the steric hindrance. So this effect can be restored by 2' fluor substitution because fluoro group acts as a hydrogen bond acceptor. So these results demonstrate that this additional hydrogen bond actually contribute to the AUA decoding. So in the case of AGM2C, similar tendency was observed, but it's not significant. This is because codon recognition by AGM2C is further supported by additional hydrogen bond with the ribosomal residue. So this anchoring interaction is redundantly stabilized by mRNA and ribosomal RNA. So anyway, this is a first reported instance of tRNA modification that recognize mRNA residue outside the codon. Okay, so I'm moving on to the final topics. It's about Kyoshin story. Kyoshin is a unique tiered modification because it has a 7 layers purine core structure, having a bulky side chain with cyclopentane diol group. So Q is present in the anticodon of tRNAs, widely distributed in bacteria and eukaryote. So last year, actually last November, we had a tRNA conference in Kanazawa, Japan. So we had 371 people from 29 countries. Maybe some people here also enjoyed this conference. So at this conference, I heard a very interesting backstory about the naming of Q from Yuki Yokoyama, who was one of the keynote speakers. I just assumed that the naming of Q simply comes from the word question or query, but it's not true. So Q was discovered by Susumu Nishimura's group in 1970s. Susumu named it from Japanese famous manga character Kyutaro, because Kyutaro is a ghost and quite different from a human child. So Susumu named it from Kyutaro. So many theater people got excited about this story. So anyway, this is a biogenesis of Q and its derivative. So in vertebrate, these 40 species have Q or glycosylated Qs. So unique to this modification, the free base of Q, called queuing, is introduced by unique base replacement reaction from G to Q, catalyzed by QTRT1 and 2 heterodimer. Since we eukaryote cannot synthesize a queuing de novo, so we need to obtain queuing as a micronutrient from gut microbiota or food. tRNA histidine and asparagine have Q, and the tRNA tyrosine Q is further galactosylated to form GalQ, and the tRNA aspartate Q is monosylated to form ManQ. However, the glycosylation enzyme for these two pathways were not known for nearly half a century since their discovery. So recently, we identified these enzymes and named QT-GAL for GalQ formation and the QT-MAN for ManQ formation. Because QT-GAL was identified by my female student, Xue Xiao, and the QT-MAN was found by male student, Dima. You know, it's perfect gender balance. So in my laboratory, a student who discovered enzyme or gene has a naming right for it. So actually, Xue asked me to name it QT-GAL, so Dima had no choice to name it. Anyway, I'm very proud of their great contribution to this project. And then thanks to productive collaboration with Shintaro Iwasaki's laboratory in Riken Institute, we performed ribosome profiling to examine the effect of Q glycosylation on decoding efficiency in a series of knockout strain versus wild-type cell. So then we compared the ribosome occupancy of all sense codons between knockout and the wild-type cell, but they are very, very complicated data set. I want to summarize the result in the next few slides. In the case of DNA tyrosine, once a QOSIN is introduced, UAU decoding is facilitated, while USC decoding a bit slows down. So QOSIN modification facilitates the decoding of UN encodon, but slows down the CN encodon. Then Q is galactosylated. UAU decoding does not change, but USC decoding slows down significantly. So Q galactosylation is a negative regulator for USC decoding. In the case of tRNA aspartate, once QOSIN is introduced, GAU decoding speeds up and the GSC decoding slows down. Then Q is monosylated, both GSC and GAU decoding slow down significantly. So Q monosylation is a negative regulator for both of cognate codons. Optimal translation is required for proper folding of the nascent proteins. Once optimal translation is disturbed by loss of tRNA modification, protein misfolding is induced and the proteostasis is affected, leading to cellular stress. To test this speculation, we used an aggregation-prone reporter and clearly observed aggregation puncta accumulated in a series of knockout strains. Then the result was compared by aggregation index as shown here. The protein aggregates are highly accumulated in all three knockout strains compared to the wild-type cell. So protein folding is impaired by loss of Q glycosylations. In other words, Q glycosylation maintains proteostasis. To visualize structural details of codon-antacodon interaction mediated by Q and glycosylated Q, we conducted cryo-EM single particle analysis of human ADS ribosome complex with tRNA and messenger RNA. We obtained a high resolution structure of the ribosome complex with different tRNAs and codons. The resolution was good enough to model the tRNA modifications. So when we look at the decoding center at the S site, we can clearly see codon-antacodon helix and the surrounding ribosome RNA residue, which tightly recognize this helix from the minor group. So this is the closed conformation with the small subunit. So in the case of QOC modification, the cyclopentane moiety of Q recognize the codon-antacodon helix from the major group through hydrogen bond network. Actually, this moiety recognizes first and the second base pair of the codon-antacodon helix. So cyclopentane moiety acts as a major group binder for codon-antacodon helix. The effect of the major group binding is stronger when Q pair with U, but weaker when Q pair with C in the codon. This observation can be explained by the base pairing geometry. So this is a morphing animation from QU to QC. So from QU to QC, the Q base moves toward major group by 1.9 angstrom, so that a cyclopentane moiety detached from the major group in QC pair. So this is the molecular basis how Q facilitates the decoding of UN encodon, but slows down the CN encodon. And their cryo structure of ManQ and GalQ recognizing UN encodon. ManQ and GalQ also works as a major group binder as shown here. So what is the most striking finding in this structure is the rotation of the cyclopentane moiety by the glycosylation. This rotation modulates the major group binding of the cyclopentane moiety with loss of the hydrogen bond network. So this is the reason why Q glycosylation slows down the decoding, especially for C and D codon. So these structure observations nicely explain our ribosome profiling data. So we are now working on mouse models of deficient Q glycosylation to look for phenotypes related to brain function in mammals. So we also have we already have a knockout mice of QT-GAL and QT-MAN in addition to QT-LT1 and 2. And then we confirm the absence of the target tRNA modification by mass spec. So all knockout mice are viable and fertile, and we don't see any developmental abnormalities so far, but we noticed heavier body weight of QT-man knockout mice in both males and females compared to the wild-type mice, whereas no difference in body weight was observed in QT-gal knockout mice. So QT-man knockout mice may have some metabolic problem. To study physiological phenotype of deficient Q glycosylations, we studied behavioral test battery for a series of knockout mice in collaboration with Miyaka's laboratory in Fujita Health University. So this is a combination of several tests involving major behavioral categories, such as anxiety-like behavior, motor function, activity level, and learning memory functions. And they are to this end, we prepared 20 males and females of both wild type and knockout mice from the same litter mate. The experiment is still in progress, but I'd like to share some of the results. So this is a light to dark transition test to examine anxiety-like behavior using two connected chambers, light chamber and the dark chamber. The mouse freely explored the two chambers for 10 minutes. Then we record their movement. And the mice naturally prefer darkness, but healthy mice briefly explore the light area. However, anxious mice avoid the light area. So this is a result of QT-GAL knockout mice. Left graph shows distance traveled in the dark area, while right graph shows time spent in the light area. As you can see, we don't see any difference in the distance traveled in the dark area between wild type and knockouts. However, QT-GAL knockout mice spend less time in the light area in both males and the females. So QT-GAL mice show anxiety-like behavior. So this is an open field test to evaluate locomotor activity. Mice freely run in a box for 2 hours. Total distance traveled is recorded at 5 minutes intervals. So this is a result of QT-man knockout mice. Curiously, we observed a female-specific phenotype. For male mice, we don't see any difference in the distance traveled between wild type and knockout. However, female knockout mice traveled distance longer than wild type mice, indicating that QT-man female mice show a hyperactive phenotype. So we want to elucidate molecular basis of this interesting phenotype in our future study. Okay, so this is a summary of the last part. So we identified two glycogen enzyme, QT-GAL and QT-MAM, and the Q-glycosylation contributed optimal transmission and proteostasis, and the cryo-EM structure analysis revealed that molecular basis of codon recognition mediated by Q-glycosylation. And the QT-GAL knockout mice showed anxiety-like behavior. QT-MAM knockout female mice show hyperactive phenotype. And finally, I'd like to thank people in my laboratory, especially Yuriko Sakaguchi and Kenjo Miyauchi for mapping of the cytoplasmic TLR modification, and Naho Akiyama for structural study of cytidine modification, and Shue Shao, Dimar, and Kensuke Ishiguro for the Q-glycosylation study, and Atsuya Ogisa and Eri Tsuruta for mouse study, and other people who helped their work. And this is a close collaboration with Matt Waldor and Satoshi Kimura for the cytidine modification, and Shintaro Iwasaki's laboratory for ribosome profiling, and Takeshi Yokoyama and Mikako Shiro's for crime study, and Miyaka's laboratory for mouse behavioral battery, and Fan Wei and Yusuke Hirabayashi for our fruitful collaboration of the mouse work. And finally, I'd like to thank our funding sources. So thank you very much for your attention.
All right. Wonderful. Wonderful talk, Tom.
Thank you.
And for people who want to ask any questions, please use the best way to do it is to use the Q&A box. If you don't see it, if you don't see the button, it's probably under the More, M-O-R-E button, and then you can click the Q&A. You can also use the chat button, but the Q&A is better because then the speaker can, if we don't answer all the questions, the speaker can address some of those questions. But I'd like to start, Tom. This is,
Mhm.
the glycosylation was
Right,
particularly amazing to me, in part because there is an independent line of research about glycoRNAs.
right, right.
In principle, I think this is the first glycoRNA whose structure was identified. But have you explored whether the glycoRNAs that people are seeing and sometimes people are saying might be exported or put on surfaces or something like this, have you
Yep.
explored whether they may actually derive from this quiasine pathway and this quiasine modification?
Right. So, actually, Kyosin glycosylation is, the monomer, right? So, galactose or mannose is attached to the Kyosin side chain, but the glycoRNA long side chain is attached to the ACP3U and other modification, right? And it's not related to the translation. It might be involved in the signaling pathway. So I'm also curious about the glycoRNA, but it's not related to the Kyosin glycosylation.
Well, what I was thinking is that the glycosylation that you see can further be extended off
Oh,
the, those initial monomers, those sugar
okay,
monomers. And in the studies where they're finding the ACP3U, it only
okay,
it's a small fraction of all the glycosylation that they see
right.
in RNA. They show that that was a critical modification, but it wasn't necessarily the major form of the
Hmm.
glycoRNAs in the cell. So that's why I was thinking about this as the starting point for
Okay,
further sugar modification.
okay.
Let me
Actually,
go ahead.
we didn't detect any long side chain attached to the transfer RNA so far. Mm. But
Okay.
mm,
And okay, so
yep,
let me ask some questions from the audience. I think one of the questions that people are asking is, you have these modifications which recognize tRNA and modify tRNA, but they could in principle recognize tRNA-like structures elsewhere.
okay.
And so Jianbing Mu was asking whether these pathways are affecting other RNAs with tRNA-like structures.
Yeah. It's very interesting question. So if there is a tRNA-like structure or stem loop structure, it looks like a tRNA anticodon region. It might be a target of some tRNA modifying enzyme, right? So yeah, it's worth to try to look for some tRNA modification in messenger RNA or other non-coding RNA.
Yeah, I definitely think that'll be interesting. We're going to do one more question just in the interest of time. So Stefanie Kaiser was asking, um, how different is the modification profile among isodecoders? And so, and she says, are there any examples where modification profiles deviate largely between different isodecoders?
Yep, yep. So this is great questions. So actually, it's very difficult to separate isodecoder, right? So like a one base mismatch or two base mismatch, it's very hard to separate. But when we analyze the RNA fragment, in the fragment basis, we can actually detect each of the isodecoder. So in the purified sample. So yeah, there are difference in the modification profile, but yeah, actually it's very difficult to
Yeah,
separate each of them, right?
it'd be very interesting if different isodecoders with different profiles
Right,
differentially expressed, like one in brain, one in the other tissues to
right,
actually see
exactly,
what expression.
exactly.
Just very quickly, Francisco, Aaron Cruz was asking, you emphasize a lot about the modifications in the anticodon region, but when they're outside of the anticodon region, what are they doing? and do they participate in decoding or potentially other functions altogether?
Yeah, so this is an open question, right? So there are many modifications in the body region, right? So if the some of the body modification is missing, so it has a phenotype, right?
Mhm.
So, but we don't know what happens. So maybe some body modification is required for the tRNA stability and also function in the translation. But
Yeah,
yeah, actually, it's open question.
yeah. A lot, clearly a lot more work needs to be done. A lot more research needs to be done.
Yeah,
So,
yeah, I
Tom,
think
I
so.
want to thank you so much for beginning our symposium. This was a really
Thank you
exciting
very
uh,
much.
seminar. If people have questions, they can continue to put them in the Q&A box.
Okay, okay.
And if Tom has time, he'll be able to answer it. It's very late right now in Japan, but
Yeah.
you can give it a shot. It's also late for our next speaker at Wuhan University in China, who is Bi-Feng Yuan. And so Bi-Feng Yuan's talk is entitled Exploring RNA Modifications Through Mass Spec and Their Correlations with Disease and Environmental Exposure. So with that introduction, I'm very excited to introduce Bi-Feng Yuan.
Yes, thank you, Dr. Jaffrey. Okay, let me share my screen. Okay.
Mhm.
Okay.
It says it's starting. It started screen sharing. We don't see it yet.
Okay, can you see my screen?
Not yet. Um, unless, yeah, now we can see it. Okay, so everything looks good.
Okay. Hello, everyone. I'm Bi-Feng Yuan from Wuhan University. I would like to thank the organizing committee for the invitation to attend this RNA mass spectrometry symposium. Today, I would like to talk about the exploring RNA modifications through mass spectrometry. The title is a little bit different from before because I'm afraid I don't have time to talk about the correlations of RNA modifications with disease and environmental exposure. So I did my postdoc research at University of California, Riverside from 2007 to 2010 under the supervision of Professor Yinsheng Wang. He will also give a talk later. After returning to China, I began to work on DNA-RNA modifications, primarily using mass spectrometry and sequence analysis. Since 2018, I have been working on metabolomics, and since 2023, I have been working on exposomics. Today, I will making report on our work related to using mass spectrometry to analyze RNA modifications. So there are many modifications on DNA and RNA. Currently, over 10 types of modifications have been identified on DNA, and more than 160 types of modifications have been discovered on RNA, as mentioned by Dr. Suzuki. So RNA modifications are closely related to the occurrence and the development of many human diseases. In our research on RNA modifications, we primarily focus on three areas. We carry out identification studies to address the questions of what modifications exist in RNA. We carry out sequence studies to determine where these modifications occur, and we explore how modifications are formed and erased. Today, I will focus on the first issue, identify RNA modifications using mass spectrometry. Here is the typical process for analyze RNA modifications. Use mass spectrometry. RNA is isolated from cells or tissues, then enzymatically digested into nucleoside. Next, a standard curve is constructed, followed by qualitative and quantitative analysis. use mass spectrometry. There are several challenges in analyzing RNA modifications use mass spectrometry. First, the purity of the isolated RNA. Second, the stability of RNA modifications during the enzymatic digestion process. And third, the detection sensitivity by mass spectrometry. Obtaining high purity RNA is essential for the accurate analysis of RNA modifications. In this work, we develop a purification method to obtain the high purity messenger RNA. First, we remove the RNA smaller than 200 nucleotides using a size selection method and achieve high purity messenger RNA through 2 rounds of polyisolation. We observed that after the second round of poly purification, the tRNA specific modification of I6A was almost undetectable. This indicates that this method can effectively remove tRNA and high purity mRNA can be obtained. Subsequently, we can accurately determine M3C in messenger RNA. In addition, we employed a method that combined poly purification with gel purification to obtain the high purity messenger RNA. We first isolate messenger RNA from total RNA use the poly purification method, and then further purify it to obtain the high purity mRNA through gel purification. Use this method, we identified 2 new modifications in yeast messenger RNA, including inosine and 2' or methylinosine. Similarly, we established a purification method for extracting high purity small RNA ranging from 16 to 28 nucleotides. First, we precipitated small RNA from total RNA using 5% PEG 8000 combined with 0.5 molar sodium chloride. Then we separate and collect RNA with length from 16 to 28 NT using HPLC from here to here, followed by enzymatic digestion and mass spectrometry analysis. With this method, we identified many new modifications within the small RNA ranging from 16 to 28 NT, showing this blue circle. In addition, we also identify a double methylation of adenosine, M1,6A, in mammalian tRNA, where M6A and M1A are widely present in RNA. We have been considering the existence of double methylation modifications at both position 1 and position 6 of adenosine in RNA. We extracted different types of RNA and analyzed whether M1,6A was present in these RNA samples. By comparing them with standard reference, we found M1,6A was present only in tRNA. The identification of M1,6A was also confirmed by high-resolution mass spectrometry analysis. After identifying M1,6A in tRNA, we carry out further analysis across different cells and tissues. We detected M1,6A in various tissues and cells that indicate its widespread presence in mammalian tRNA. We further analyzed the presence of M1,6A in individual tRNA molecules. We isolate glycine, histidine, and methionine tRNA through hybridization with biotin-labeled DNA probes and found that M16C was detected in all these tRNAs. Obtaining high purity RNA is crucial for analyzing RNA modifications. Additionally, suitable enzymatic digestion conditions are also important for the accurate analysis of RNA modifications. Typical RNA digestion conditions generally involve using the nucleus P1 and alkaline and for digestion in a pH 5.6 solution, followed by further digestion with the phosphorodiesterase 1 and the alkaline phosphatase at pH 8.9. However, some RNA modifications are not stable enough and are sensitive to acidic or alkaline conditions, which may lead to the degradation or conversion of RNA modifications. For example, M1A can be converted to M6A under alkaline conditions, leading to the inaccurate analysis. We developed a neutral enzymatic digestion conditions that is carried out in a single tube, maintain a constant neutral pH 7.0, which can effectively avoid the degradation and the conversion issue of RNA modifications. We observe that the levels of normal ACGU base obtained with neutral enzyme digestion conditions are consistent with those from the traditional digestion method. However, for RNA modifications, the neutral enzyme digestion method can achieve higher signals for many of these modifications. Using this method, we detected 3 types of hydroxymethylated RNA modifications in mammalian RNA. These hydroxymethylated RNA modifications are generally not stable in acidic or alkaline conditions. They are not easily detected with using traditional digestion conditions. Additionally, using the neutral enzymatic digestion method, we also detected hydroxymethylated adenine in mammalian DNA. This is the standard reference for HM6DA, and the peaks shown here is the HM6DA detected from HAG293T DNA. Moreover, a high-resolution mass spectrometry of the HM6DA detected from genomic DNA is consistent to the standard reference. Furthermore, HM6DA is present in various cells and tissues. RNA modifications are generally present in low abundance in RNA, so the sensitivity of the analytical method determines the ability to detect RNA modifications. Here, we establish a method that combines chemical derivatization with mass spectrometry analysis. This derivatization reagent can contain reaction group that can specifically react with the modified base, allowing for the labeling of the modified base. This process adds enrichment group and MS sensitive group to the modified base, enable subsequent specifically enrichment of the modified base and the enhanced detection sensitivity during mass spectrometry analysis. Here is an example. After chemical derivatization with SAMCT reagent, the detection sensitivity of uridine by mass spectrometry was increased by 1,400-fold. This is primarily due to the presence of a permanently positively charged quaternary ammonia group on the SAMCT, which increased the annihilation efficiency in mass spectrometry analysis. Using the sensitive detection method, we identified 5-methyl uracil in mammalian messenger RNA. here are the chemical derivatization reagents we have developed over the past years for the high sensitivity mass spectrometry analysis of DNA and RNA modifications. So here, we summarize the method for analyzing RNA modifications in 2 review papers, and the method mentioned in my report today can be found in these reviews. Okay, that's all my report for today. Finally, I would like to thank the members of our group and the funding support and the collaborators for their support. Thank you. Thank you very much.
All right, that was a really great talk. And you know, it's really interesting about using these chemical modifying agents to detect the modifications that are otherwise difficult to detect. Do you think one problem in mass spectrometry sometimes is isobaric modifications, where the 2 modifications have the same mass? Are you finding that these chemical modifying agents help solve that problem?
To some extent, they may be so the separation of the iso modifications after chemical labeling, the elution time may change for the different modifications. Yeah. For example, previously we detected that's a carrier molecules after derivatization. So the carrier molecules, the 2 carrier molecules can be separated in the HPLC.
Okay. Let me ask a question from the Q&A. And so Yinsheng Wang was saying, did you check the stability of 5-HMRC and the ribo version of HMC and ACCM in acidic and basic conditions? He says you observe much higher levels of these modified nucleosides with neutral digestion. So he's wondering about the stability of these modifications.
HM5C is relatively stable. The hydroxymethylated modification occur in the nitrogen for HM6A and HM2G. These 2 are not stable. Under the acidic or alkaline conditions, HM5C is stable.
Got it. So which are the modifications that really show the instability in either acidic or basic conditions? You brought up M1A, but most of the time when people study tRNA, you don't see the Dimroth rearrangement and see M6A. Most people don't see M6A in their tRNA samples. So which modifications are the most susceptible to the acidic and basic conditions?
For example, just like the hydroxymethylated, these two are not stable, and maybe there are some other modifications, but we didn't check all of them.
Got it. So just those 2 are the ones that we've seen so far.
Yeah. Yes.
Got it. Okay. Very interesting. Are there other questions from the group? All right. Then Bi-Feng, thank you so much. It was a really interesting presentation. I look forward to seeing more of these functions of the 1,6 dimethyl. So this will be very interesting. Now with that, I'd like to thank Bi-Feng, and we'll switch over to Stefanie.
Okay, thank you.
And I've known Stefanie for many years. She's helped us with various projects. And so Stefanie comes from the Goethe University in Frankfurt in Germany, and her talk is Hunting for Truth Towards More Accurate RNA Modification Stoichiometry. And with that, we'll hand it over to Stefanie. And just remember, please use the Q&A button for questions. And if you don't see the Q&A button, you might have to click the More button, and then you'll see the Q&A link. And Stefanie, you should see a timer, and you'll hear a ring at the end of your when your time's up. All right, Stefanie, take it away.
So you guys can see my slide, I assume.
Yep, it looks great.
Fantastic. So thank you, Samie, for the nice introduction, and thank you for the invitation to speak here. So I will basically pick up at the same point where we just stopped. So there will be a lot more information on these little kind of things that disturb mass spec analysis when it comes to modifications. So before I go more deeply into this, if you look at mass spec of RNA modifications, there's basically two ways of quantifying the abundance of RNA modifications. We do this to understand biological processes, and very often it's enough to do a relative quantification. So to learn basically, if I compare one sample with another type of sample, is there more or less of a certain modification? For example, if you look at a mouse and you want to look at messenger RNA, you want to learn, is there more M6A in the liver compared to the brain, or is there more M3C compared brain and liver? So you can do a relative quantification, and this would, for example, for mice actually look like this. And this looks very impressive. So there's a six-fold more M3C in the mouse brain messenger RNA. Now, if we look at absolute quantities, the picture is, I mean, technically the data is the same, right? But you will see that of course, you all know that M6A is way more abundant in general in messenger RNA, right? While M3C is very low abundant. In fact, for liver, it's very close to the limit of quantification, while in the brain, then it is a little bit higher. But the point I want to make here is that sometimes it's very important not only to report relative changes between samples, but also have a good idea of the numbers of what you're actually talking about. So how stoichiometric is it? And now the problem is, if you want to do something like this, an absolute quantification where you really put numbers on your biological data, the question is, are these numbers actually true? Are they accurate? And for this, we first need to talk briefly about truth and what it means in absolute quantification. And of course, you all know what precision is. It's basically standard deviation, right? You can do an analysis, and here the standard deviation is very high. Yeah, the data points are very far from each other. And in analysis 2, the precision is better. Yeah, data points are close to each other. Yet this analysis does not tell us anything about hitting the true value. For this, we need actually the true value, which is here in the center of this board. And now you will see, okay, both of these analyses are not very close to the true value, although their precisions are different. So what can happen in an analysis is that the precision is not too good, but still on average, you will hit the true value, or the precision is actually really good and you hit the true value. And if this happens, this is what we call high accuracy. So if we have a high precision and trueness, we have a high accuracy. And I think this is the goal, or this is what we want in the end, because only if we have a really good accuracy, we can interpret the biological data that we get from these analyses. So how do we get there? So what my lab is doing when it comes to RNA mass spectrometry is, today I want to focus on this bottom-up approach we had so far, so digesting them down to the nucleoside level and then quantifying them with the mass spec. And some 10 years ago, when I was still in Mainz with Mark Helm, we figured out, okay, we want to quantify RNA modifications in various samples. And since we are analytical chemists, kind of, we were like, okay, in UV measurements, this is very easy because you can just correlate the analyte concentration with the signal intensity using the extinction coefficient, and that's it. Yet RNA modifications are low abundant, so you cannot use UV quantification, so you need to use the mass spectrometer. And for mass spectrometry, this correlation of concentration of analyte and signal intensity is not as clear. So basically, all sorts of ionization buffer elution times impact the signal, as does the instrument settings themselves. And then in the worst case, even the sample parameters, like an isolation you have done on one day and an isolation you've done on another day, you have different salt loads in the samples. And then your signal does not correspond to the analyte concentration anymore. So back in the day, we were like, okay, we have to fix this. And we came up with this known concept in analytical chemistry known stable isotope labeled internal standards. Basically, you take a modified nucleoside, and what you do is you ask bacteria or yeast, your friendly neighborhood helpers, to incorporate stable isotope labeled atoms, for example, 13 carbon or 15 nitrogen at all positions. So you get this internal standard. You isolate this from the bacteria or the yeast, and then you spike it in first into your weight standard of the modification when you do a calibration curve, so you can correct for any fluctuations. And you also spike it into your sample. So you get a good correlation of your sample to this internal standard. And with this, you can then really calculate the amount of a modification in moles. And with this, we are ready to do absolute quantification. And this is a long time ago, so this is running quite nice. So the workflow that I think most of you people are doing and that everybody's doing, you have some sort of a sample isolation. Most of the time it's triazole isolation and then an ethanol precipitation, followed by some sort of purification for the RNA type you're interested in. In the next step comes the hydrolysis, which can be done with different kind of hydrolysis enzymes. Then there is a sample preparation step. Sometimes it's very smart to filter the sample to remove the enzymes. Sometimes it's not the best idea to filter them to remove the enzymes because some of the modifications actually get lost. I will get into this in a moment. Then the next step is the calibration, so weighing in the synthetic standard, making serial dilutions, and then injecting that together with your samples into the LC-MS, followed by the data analysis. So this is basically the workflow that most of us do. And I want to briefly touch on a couple of areas where we have already found problems in the workflow that can really change the outcome of the results in the end. Starting with the isolation step, you all know that our total RNA is usually a lot of ribosomal RNA, which is really nice if you study ribosomal RNA. But if you study tRNA, messenger RNA, or small non-coding RNAs, it's quite troublesome. And now I would just want to advertise a paper that is not from me, but actually from Mark Helm, but I think it's very, very important to this crowd, is that here the integrity of the RNA during the isolation can dictate basically your downstream success of purification and your results. So what they have actually described is that there is certain modifications like M3U, M66A, and AM, that if these modified nucleosides show up in your, let's say, tRNA analysis, it indicates that your tRNA is contaminated with ribosomal RNA fragments. And the ribosomal RNA fragments do not only contain these modifications, but also canonical nucleosides. And since we usually normalize to canonical nucleosides, your whole tRNA modification quantification can be obscured. So this is something to be aware about. The second step, we already talked a little bit briefly about this, is the hydrolysis. So basically, there's all types of digestion protocols out there. There's even commercial kits that are really good. Then there is the very classical approach from the 90s. And then there's an approach that I prefer myself, which I call the Didon lab approach, because I learned it when I was a postdoc in Pete Didon's lab. All of these approaches contain nucleases, phosphodiesterase 1 and alkaline phosphatase, or at least I think they do, because for the NEB one, we do not really know. But the thing is, if we compare these protocols, what you find is for many modifications, you get exactly the same numbers, but for some, you do not. And this is something to be aware of, because it basically means if you compare sample to sample within the same kind of experiment, it doesn't really matter. But if you compare samples you have analyzed half a year ago and you used a kit versus then a homemade kind of digestion cocktail, you might run into big problems. So this is something to be aware about. The second thing is that usually these enzymes are contaminated with deaminases, which means that C is converted to U and that A is converted to inosine. And there is deaminase inhibitors, pentostatin or tetrahydrouridine, that you can add to your digestion, and then you will get probably the more accurate results because you stop unwanted deamination of these nucleosides. All right, the final step before I go into the calibration would be the filtration. So the filtration is usually done to get loss or get rid of the enzymes. And unfortunately, the membranes that are used in these tubes that we use for filtering can lead to the loss, as you see in this heat map. Here you see a loss of M66A or I6A, and inosine are lost from the polyether sulfane membranes. But not from composite carbon membranes. So it really matters which kind of filter you use before filtration and then before injection into the mass spec, because it will change your quantitative results in the end. So this is something to be aware about. Okay, so the next step that we have not yet at least published anything about, but I want to go into more detail because this is something we have been studying for the last year. Basically, if you want to have true accurate results, everything hinges on the calibration and everything hinges on the purity of the synthetic standard and its stability once you have prepared it in aqueous solution. And the thing with the purity, basically what all of us do is we go to our vendor, let's say vendor X, we buy the modification of interest, and then they come with a certificate of analysis that tells you, okay, this is 97% pure. So you prepare the solution and then you measure with that and quantify with this. The thing is, actually, this is not a quantitative purity. I asked the vendors, and the reply I got is that these certificates of analysis is not meant for quantitative analysis, but actually for qualitative analysis. So the numbers that they give are actually not true, which then, of course, challenges everything we are doing when we're calibrating, right? So how can we work on this, right? What can we do about this? And one idea, oh yeah, this is an example, for example. I recently bought a batch of 1-methyladenosine, and we already know about the Dimroth rearrangement, right? And what you can see in this fresh solution that we just bought, we already have 7% abundance of M6A. So just mentioning contamination is really a problem a lot of times. Now let's come back to the point with the purity and how we can determine this. One solution is actually to do quantitative NMR, because quantitative NMR depends mainly on the signals of your modified nucleosides, as you can see in this NMR spectrum. And if you weigh in the standard and you spike in a calibrant that has a known purity because it's a primary standard, you can run calculations based on the intensity of these proton NMR signals, and you can come up with the purity of these modified nucleosides by putting in all these values that you get from the NMR. With this, you can then correct the purity that you have, and with this, you can then run actually well-calibrated absolute quantification experiments in the mass spec. So how does this work? So basically, the first thing we did is we first quantified the standards themselves. So there's 3 common standards that are used for qNMR analysis. And what you can see is that the standard deviations, if you quantify each other with the one of these other primary standards, is that the deviation is very, very small. So then we did this in the next step with a bunch of modified nucleosides that we had in sufficient quantities. And you can see that in general, the purity is quite good, especially M5U is close to 100. M2G, not that good, just 90%. But now we know about this, so we can put this into our considerations when doing quantification. And now the important thing is, or the interesting thing is, just imagine for a moment that you have a modified nucleoside of unknown purity that contains, let's say, salt contamination. The salt contamination you would not be able to see in the LC UV chromatograph, LC-MS chromatogram, or in the NMR spectrum itself. So you would not even know that the salt is present. But with the qNMR, you would see that your expected purity deviates from the 100% purity you expect. So one example, what we have done is we took M5U, which was close to 100%. We spiked in 25% of salt, just sodium chloride. And as you can see, we then quantified again the purity, and we got actually the 75% as expected. So even if you do not know how pure your standards are, you can be sure even salt contaminations will not make you a difficult life. So if you have enough of this material available, go for qNMR analysis. All right, now the first step is done. We now know how pure the synthetic standard is. Now we have weighed it out and made solutions, aqueous solutions. And actually water is a very reactive chemical reagent, which means that some of the modifications might do chemical reactions. And we wanted to determine how stable are modified nucleosides in the calibration solutions. And for this, we came up with this workflow. So we weighed in 40 to 50 modified nucleosides in single tubes and triplicates. We dissolved them. We diluted them to a concentration that is useful for LC UV MS chromatography, distributed them on roughly 120 plates. And then these 120 plates were sealed and they were put at -80 degrees, -20, 8, 20, and 40 degrees Celsius for 1 to now 6 months, but it will go on up to 36 months. At time point 0 and after every month, we took one of the plates. We spiked in internal standards to catch deviations in the UV detection, and we did LC UV MS analysis. What we expect is an unstable modification should show a loss in the UV signal of the peak, and with the MS scan that we do in parallel, we can figure out the products that emerge in case of an instability. So this is what we've done, and now just a brief overview of what we see for, let's say, stable modifications, and I can calm you down. Everything is fine. Most of them are fairly stable, even at room temperature for up to 6 months, but not all of them. There is actually 3 that show a higher abundance the longer you keep them standing, which argues towards some sort of adsorption effects that then get into some sort of equilibrium at later stages and release. This is something we have to look into, but this is very worrisome, in fact. And then there are some really unstable ones that degrade over time in a temperature-dependent way. I want to give you an example that I think most of us can relate to is acetylated cytidine. And what you can see here is that at -20, -80 degrees, it's very stable. We have a little bit of an evaporation, so the values are actually rising a little bit. But at room temperature, you see that it goes down with time substantially. So what is going on with AC4C over time? If we look at the chromatogram, at the beginning, we only have the black chromatogram of AC4C in the internal standard. And what you get with time is 2 additional peaks up here. And one of them, according to the mass spec, is cytidine, and the other one is uridine. So basically what is happening is it's deacetylation and then a deamination reaction, probably due to the acetic acid that is being made from the deacetylation reaction. So this is something to be aware of, because this means that these standard solutions really need to be stored properly for probably not too long time, but we don't know yet. So the take-home message of this talk is that the quantitative analysis of RNA modifications by mass spectrometry requires substantial validation and more validation to, in the end, reach our goal, which is getting biologically relevant data. And what I think is now very clear to everybody as well, we need some sort of international harmonization, because along this workflow that all of us are using are so many potential pitfalls, artifacts, some deviations that will change the results. And this international harmonization will actually happen. There's the first workshop of the Human RNO Project Consortium in August of this year. Some of you have already registered for this workshop. And the goal of this workshop is to define the alphabet of the human RNome, basically using mass spectrometry of RNA. The program you can see here, and the whole idea of the Human RNome Project as well. I just want to focus on the program for a moment. What we have is a full week where we go to the labs and we do experiments on instruments, and we come up with a harmonized protocol so that in the future we all get hopefully the same accurate results. We also have talks and perspectives, and we have breakout sessions with a lot of discussions. What is the future of the human RNome, and what should be our next steps to continue? So with this, I am at the end, and I want to thank you very much for your attention.
All right, Stefanie, that was wonderful, and people should feel free to ask any questions. You know, the AC4C is particularly interesting because there is, this debate about the relevance of AC4C in mRNA, and some groups are saying that the AC4C is not present, and I can't help but think that maybe sometimes C to U editing in mRNA, which people might think is due to an ApoBec, could have actually been due to conversion of the AC4C to U during sample processing. So have you guys considered about the AC4C stability in mRNA accounting for some of the differences that people have seen in the AC4C measurements in the transcriptome?
Well, I would, after we have now this data, assume that it might have something to do with it. We did some calculations as well with a theoretical chemist, and as a matter of fact, AC4C is one of the most unstable modifications. So I would assume that this is something that can happen either within the cell or at any step of the isolation. So that basically the different results that we see in mRNA might actually occur in the process of the isolation or purification, because it's temperature dependent, right? So, and probably pH dependent as well. So I would guess, yes, this is something we need to be all very careful about, especially for this modification and a bunch of others.
Okay. And I want to ask another question. I think we have a little bit of time. Harmonization sounds like a very important thing that you want to do. But if you're dealing with a stable modification, one of these modifications that's not changing their stability, wouldn't simply using an internal standard, which is isotopically labeled or some other way to get quantitative accuracy, wouldn't that alone be enough, even if someone uses one type of filter in Japan and one type of filter in America and one type of filter in Germany? You could have different losses, and different precipitation approaches could lead to different amounts, but the internal standard controls for all of that variation. Wouldn't that be correct? And only the harmonization is more important for the unstable modifications, like the thio modifications, the AC4C, and things like that. Is that reasonable or no?
I would say the harmonization is an issue that involves all steps of the process, including the mass spec, actually. So I think it's very important to give harmonized advice on all types of analysis, including really ranging from sample preparation to the end. You bring in a very good point, and that is, at what point is the internal standard spiked in? Because this can control for many artifacts that might happen along the way. But yet stabilities are different on nucleoside and on the intact RNA level. So this is a very difficult discussion that I would guess we will have hours and hours going on in August to discuss when and at what point to do this best. Because, as you say, we might catch a lot of problems with this kind of approach.
Okay, let me go to some of the questions because I think I don't think I've monopolized all the time here. So Justin Dingman is saying, great talk. I'm curious why you prefer the Didon lab method of digestion versus others. What are the pros and cons of the Didon lab method? He says, in my hands, yields are generally much lower using this method than the 1990s method, which can greatly impact the limit of detection and limit of quantification for samples. So he says he thinks about this all the time. And so it's great to get your thoughts on this.
I love that. Okay, we should have a proper discussion, having a bottle of beer in our hands. So my reason for that is it's a one-pot digestion, which means it does not require switching pH. Yeah, the 1990 protocol is done first in ammonium acetated pH 5 point something, and then the alkaline phosphatase. So you have to switch to pH 8 to 9 to do the dephosphorylation. And I think the main benefit of the one pot is that you do everything in this slightly alkaline environment. And so the stability, at least for those that are acid sensitive, they are better protected this way. But there's one thing. I think there's not one hydrolysis protocol that will answer all our analytical needs. So I think it depends on the modification you're interested in to decide which protocol you use in the end. Because sometimes I also think that NP1 or the 1990 protocol has better release. What is also very important is the 1990 protocol even works if you don't have phosphodiesterase 1 around. The benzonase protocol does not work without phosphodiesterase 1. So this is some of these little details, right, that can really screw up all of your results.
Yeah, yeah. These are very complex issues
Yes, they are.
And you're perfectly correct. So Robert Ross is asking, you mentioned that the presence of AM is an indication of RNA, ribosomal RNA contamination.
Yes.
And so he's saying, by simply saying is the lack of AM, does that mean that you have highly pure tRNA?
I personally see it this way. I mean, it depends on the organism you're studying, but in human, if I don't, or I have very low M6A, very low AM, and very low M66A in my tRNA preparation, I would guess at least rRNA fragmentation was not an issue.
But are there other contaminating RNAs that you have to be worried about, or is it ribosomal RNA alone that you have to worry about just because it's so abundant? You know what I mean? But is there anything else that contaminates the tRNA fractions that you have to worry about, which may also not have AM?
Well, everything that has the same size as tRNAs, and there might be RNAs that we do not know yet about that have a certain modification or modification tope that we are not aware of. So like a black swan theory, right? So we will learn that hopefully in the next decades to come.
Okay. All right. With that, thank you, Stefanie, so much. A really interesting
Thank you for having me.
and it makes me a little bit nervous sometimes. I also like it. I also tell people just don't use filters, but I didn't realize that it's the right filter. You have to use the right filter. I try to avoid filters in everything. But this was very interesting. The talks are going to be online soon. And so I'm going to go back to that slide and look at the different filter compositions. And
it's also in our accounts paper.
Okay.
So it's published 2023, actually not 2024, but yeah.
Okay, wonderful. Thank you very much, Stefanie.
Thank
So for the other questions, hopefully Stefanie can answer them directly. All right, so right now we're going to switch to Dr. Henning Urlaub at the Max Planck Institute, and he's going to talk about chemical cross-linking extends and complements UV cross-linking in the analysis of RNA, DNA, nucleic acid, protein interactions by mass spec. And so Henning, please, we can see your slides, so please take it away.
Great. Hello, everyone. Good afternoon or good morning. Sorry, here's afternoon in Germany. Do you see my slides in the presenting mode? And
Yep,
you see my laser pointer as well?
We see both the laser pointer and the slides. It looks great.
Fantastic. And you can hear me, I hear. Okay. Yeah, thank you very much for to the organizers inviting me to this exciting meeting. And yeah, we shift now a little bit gears because I will talk about as announced, about the chemical cross-linking between proteins, RNA, and DNA, and how we detect these cross-links. So basically, we are interested of how proteins contact RNA and DNA molecules. I would state that actually no DNA or RNA molecule is on its own in a cell. It's always complex with proteins at different time points, but these protein, DNA or RNA complexes are really essential. They are for DNA in DNA replication, in gene expression, and DNA repair, and for RNA, of course, in transcription, processing of the RNAs and splicing, and then export, for instance, from the nucleus into the cytoplasm, and then translation. All is mediated with the interaction of proteins. So we are interested of how exactly the proteins and which regions of the proteins and which amino acid contacts specifically RNA or DNA molecules. And for this one, we have established years ago a workflow, namely we perform cross-linking to fix the interaction of a protein with, as shown here, its RNA molecule. Then we digest the RNA and the DNA, and we digest the protein, and what is then left is more or less an oligonucleotide, in this case, connected still to the peptide derived after digestion of the protein. Then we have to enrich the species and then can do mass spectrometry. And with mass spectrometry, we are able to sequence the peptide moiety, and when cross-linked at a certain amino acid, we see a mass adduct that corresponds to the residual cross-linked nucleotide, dinucleotide, or trinucleotide derived from the digested RNA or DNA. And of course, when we do this at a little bit larger scale and with more complex sample, we need a certain data analysis. How do we fix or how do we cross-link our proteins to RNA? We use 4 different cross-linking reactions. We have the UV light, and the UV light introduce a covalent bond between the side chain, in this case tyrosine, of a protein to an very adjacent nucleotide or nucleotide in close proximity, and a direct new covalent bond. And because there's nothing in between, no spacer, no other molecule, this is often referred to be a zero-length cross-linker. Of course, you can use the natural UV reactivity of uracil, for instance, but you can also use 4-thiol-U or 6-thiol G derivatized RNA molecules, and these all work with UV cross-linking. There are also methods for chemical cross-linking, and here I would like to mention formaldehyde, which reacts with the adenosine, and upon building a Schiff space, then with the protein in close proximity. Another reagent that has been established already years ago and also has been evaluated by Dan Faber's lab a couple of years ago for protein RNA cross-linking is deoxybutane. It reacts with the N7 position here of a guanine and then with the protein, and you have a covalent link between the protein and the purine here with a spacer of the reagent in between. Similarly, the nitrogen mustard, it's also a very potent reagent, reacts with the N7 position here of a guanine, and then also with the protein to form a covalent link between the guanine and the protein. These are the reactions that we use, and we evaluated these cross-linking reactions on protein RNA complexes first, and for this one we used isolated ribosomes from E. coli and cross-linked them either with UV light, with deoxybutane, nitrogen mustard, or formaldehyde. After cross-linking, we digested here, in this case, the RNA with benzonase, for instance, and we digested the protein moiety with trypsin. We end up with non-cross-linked peptide, non-cross-linked RNA, but also with cross-linked peptide to RNA oligonucleotides. And very important, because the cross-linking yield is somehow mediocre, we need to enrich these species, especially, and here we use titanium dioxide. Titanium dioxide matrix has been introduced to enrich actually for phosphorylated peptides. Here we make use of the phosphate, of the phosphate, for instance, at the 3' end, or if it's a dinucleotide, also at the other phosphates. And by this one, we can specifically enrich for this cross-linked, as we call them, heteroconjugates. We also remove the non-cross-linked RNA by simply C18 chromatography, reverse phase chromatography at acidic pH. And then we eluted up from the titanium dioxide the species and performed LC-MS analysis. And this then is a little bit complicated because this is a spectrum where any amino acid could be actually modified, cross-linked, and we do not know to which nucleotide actually is cross-linked. So the first thing is we have to do a manual MS2 spectra annotation. And for UV cross-linking, this is relatively straightforward in my opinion, but we observe two different types of crosslinks. Basically in UV light, one is where the crosslink is, for instance, tyrosine to uracil here is stable, and where we also observed in the mass spec in the product ions, ammonium ions from the tyrosine carrying the uracil moiety here. But sometimes we also observe, even on the precursor level, that we have a species that has lost, for instance, or which corresponds to uracil that has lost water in a neutral loss, or to cytosine which has lost ammonia. So we can actually not distinguish here in this case after UV cross-linking whether we have a uracil cross-linked or a cytosine cross-linked. But therefore we refer to these cross-links as C or U. These are basically the two cross-linking reactions we observed after UV cross-linking. For nitrogen mustard, we observe, of course, cross-links to G, and especially with a histidine in a peptide sequence, or very also abundant with a methionine. And with the methionine, we have really a nice marker ion that resides from the methionine still cross-linked with the nitrogen mustard to the G and the corresponding nucleotide. With deoxybutane, it's similar to nitrogen mustard. We also see methionine crosslinks, histidine crosslinks, but surprisingly, we also observe crosslinks of G via the DEB to glutamic acid. For formaldehyde, it should be also straightforward, but here's a phenomenon that we observed in MS2. So upon sequencing, namely that the crosslink is labile in the gas phase. And what we observed then is a clear crosslink, or that the adenosine gets modified by Fa, so it has an additional C atom in the mass, but we do not see any adducts formed from the peptide. In other words, we can accurately determine the peptide sequence and know that the A, for instance, because of this very nice marker ion here in the lower M/Z range, contains or derived from the Fa, from the formaldehyde, and therefore we conclude definitely that this peptide sequence has been cross-linked to this A upon Fa treatment. Okay, so once we have once we were able to identify all these crosslinks, we sum up and we set out some presets. namely where we defined the sum formula of the precursors upon modification by UV cross-linking, or the same is for 4-thiol-U, for instance, derivatized uracil, or with nitrogen mustard, and also the sum formula of the respective product ions, or here I have to say the product ion mass adducts that can be present in on one amino acid of the peptide sequence. So everything was then assembled and was automatically for database search of crosslink, and therefore we developed, or they put these presets into a search engine or software tool we call NuXL in the OpenMS platform with the help of Oliver Kohlbacher and Timo Sachsenberg, and this is outlined here. So we have different presets for RNA, which is UV, 4-thiol-U, 6-thiol-G, DEB, nitrogen mustard, and formaldehyde. And for DNA, we have simply used UV, DEB, nitrogen mustard, and FA. So these are default presets. And then we can run on our data, on our mass spec data, this software tool. It works simply in MS2 spectrum processing. We have a matching step of in silico generated PSM, so non-cross-linked spectra, or in silico cross-linked spectra matches. We get the spectrum matches. We have peak annotation and cross-link localization, which are amino acid. We have a post-scoring protein invariance, and we use percolator, and we have annotated and, of course, interactive tables. And this is then shown here, the features again. So we can have ammonium ions that are modified by the cross-link nucleotide. We see shift of the product ions according to the mass adduct that sticks to the amino acid. And of course, we have the still residual cross-linker precursor ions in the MS2 spectrum, which should also show mass shift. Very important is to view the data, in my opinion, let's say, to check the data quality. And this is the MS2 spectrum of a peptide. The sequence is given here, cross-linked to a uracil via this phenylalanine. And very important is here is the ammonium ion IF plus U of the phenylalanine, which confirms the cross-linking site here. Below there is a view of a part of the table, namely the sequence that has been identified, the best localization sequence, which is the phenylalanine here, the cross-link nucleotide, and of course some other values. The same is also, this is all in the OpenMS platform, but the same is also possible in Proteome Discoverer by thermo-distributed, and there you get this table as an output. And if you click on this identified protein with this peptide, you get the sequence and the annotated sequence. And in this case, you see a Y6 is cross-linked to an uracil, so the Y6 ion and the corresponding amino acid is the cross-link 1. So this works nicely. We have established this one, and then we went larger, and we cross-linked by UV and chemicals entire E. coli cells, and this was amazing results we obtained. This is here under the panel A, the number of cross-linked proteins that we found and the number of cross-linked peptides. And very important is that both the chemicals deoxybutane and nitrogen mustard really showed a lot of cross-linked proteins as compared to UV and also compared to formaldehyde. Correspondingly, the number of peptides that we found to be cross-linked are much higher in deoxybutane cross-link and nitrogen mustard as compared to UV and FA. Regarding the nucleotides or the bases that are cross-linked, we found under UV light, as expected, most of CU. So here we cannot distinguish, as I said, between a cytosine and a uracil, but also a lot of U cross-links. In this case, barely any GA crosslinks, but this changes then when we use deoxybutane, nitrogen mustard, or formaldehyde, where we observe really a lot of G crosslinks and A crosslinks. In terms of amino acids, it's better to look here at this panel C. We see a clear preference in UV light to tyrosine, followed by lysine, phenylalanine, tryptophan, cysteine. And as I said, in DEB and nitrogen mustard, in DEB we have mainly methionines cross-linked, histidines, glutamates, and in nitrogen mustard, sorry, this was DEB, nitrogen mustard, again, methionine cross-linked. So the point is, this looks very nice. We have a lot of data and what to do with the data. The first thing what we always do is look at structures of protein RNA complexes that are available in E. coli, or where the RNA and protein structure has been solved. And the prime example is the E. coli ribosomes, in this case in the cell. And here is matched the crosslink sites of derived from UV, DB, nitrogen mustard, and FA. And here are the distance of the C alpha atom of the peptide to the next neighboring nucleotide in the structure. UV has an approximate distance in average of 6.8 angstrom. Slightly higher is DEB and nitrogen mustard. FA was not possible because of the reason I introduced at the beginning, that it's barely possible to locate the actual cross-linked amino acid. Other examples is the PNP of E. coli, which is part of the degradosome of mRNA, of RNAs in E. coli cells, and this forms a tetramer. And very important here is this cleft, where the RNA goes in, and here we observed exclusively under UV light these crosslinks. Very interesting, though there are other residues that should be cross-linkable with chemicals, but we did not find them, presumably because the chemicals are, or this cleft or this caveat is too tight that the chemicals go in. But we observed in other parts of this protein, namely in the S1 or KH domain of the protein, the crosslinks by chemicals. And this is a little bit more flexible region, but is also in contact to RNA, in this case of small RNAs. Another example is the signal recognition particle. And why have I chosen this example? Because this is a very, very nice example that you see here, namely that the chemical crosslinks are in particular able to crosslink, in this case, double-stranded RNA and also DNA, you will see later, while UV crosslinks we barely observe or did not observe at all in this region. Okay. Another phenomenon we observed is outlined here, namely that UV and chemical crosslink almost crosslinks all enzymes or proteins of the TCA cycle in E. coli. And this was really amazing. And this is shown here with a color code. But we ask ourselves, might this all be due to the abundance of the protein, or might this be due to the crowding effect, that all this is covered with RNA or single nucleotides and we cross-link them? But this was indeed not the case, although RNA binding proteins are highly abundant in E. coli, but what we found to be cross-linked are not the most abundant proteins, the RNA binding proteins in E. coli, but also a lot of proteins that has been assigned as non-binders yet. And in terms of the cross-linkable amino acids here and proteins, UV covers more these abundant proteins in cross-link as DB or nitrogen mustard does. One can also do some feature enrichment of the cross-link proteins. Let's say this is very interesting. So we see a slight enrichment of alpha helices and beta strands. We have an enrichment, of course, as expected from RNA binders, and especially of the conserved regions of proteins. When we do GO annotation, we see, of course, cytosolic, nucleic acid binding complex, and so on. Especially in domains, we see RNA binding domains, OB folds, Kolchak DNA binding domains and other domains enriched in our cross-link analysis. A puzzling phenomenon is the possible cross-link of proteins to DNA in E. coli, and I highlighted here two examples or three examples. One is the DNA binding protein integration host factor, and it has been the structure shown with DNA, but in fact, we observed all these highlighted amino acid actually cross-linked to RNA based on our mass spec analysis. Here in this DNA binding protein HU alpha, we cannot distinguish between RNA or DNA cross-link because we see an A or G adduct, and this A might be a deoxyribose, and this now the A might be a ribonucleotide, but this G might be deoxyribonucleotide, and both these are isobaric. So we have problems to distinguish between RNA and DNA on a single nucleotide level. Another interesting phenomenon was that we found very or almost all transcriptional repressors in E. coli cross-linked to RNA or DNA. And many of these repressors contains an HTH and helix turn helix motif, which is known to bind DNA, and we also found this cross-link, but we often observe cross-link in regions outside of the HTH domain. So,
interruption, Dr. Urlaub. We have 1 minute left to wrap up for the questions. Okay.
okay.
Thank you. Apologies for the interruption.
Thank you.
Thank you.
So, and of course, when we did also some benchmarking because there's other softwares around that might enable the identification of these cross-linking sites. And one of them is MS Fragger, which is really an excellent software. And they have also, is possible to do a similar analysis, and we benchmarked this one. But at the end, it was surprising. So they perform similarly. But the point is that especially for UV and DEB, we get better results or more results using our NuXL software approach. And by this one, I would like to come to a summary, namely that UV and chemical DNA RNA protein cross-linking, this can involve interaction sites up to single amino acids. We have generated a special search engine, NuXL, in OpenMS or in Proteome Discoverer, and it identifies any cross-link, irrespectively in recombinant complexes or in entire cells. We show that chemical cross-link extends UV cross-linking, and of course in chemical cross-link, adenosine and guanosine are preferentially cross-linked. And what we could not show now is that sequencing of cross-linked RNA moiety is still very challenging for us, but it should be possible, and it is possible. And by this one, I would like to acknowledge my co-workers, University of Tübingen with Oliver Kohlbacher and Timo, and Aslan for helping setting up NuXL, Thermo Fisher for the support and the implementation of our search engine in Proteome Discoverer, and for the bioinformatic analysis, Juliane Liepe and her group at our institute. So thank you for your attention.
Henning, this was such a wonderful talk. And if we could have the atlas of every protein and the RNA binding surfaces on every protein, yes, this would be incredible. And, it will just be so useful, especially because, Matthias Hense and others have said that there's a lot of moonlighting proteins, and being able to independently validate it using this approach is incredibly valuable. What are the barriers that you see to get a mammalian data set rather than an E. coli data set? Because I work with mammalian cells, and I want to know all the mammalian proteins.
In mammalian, it is also possible, but we are faced with some problems. First of all, the enrichment strategy. We use titanium dioxide, and mammalian cells contain a lot of phosphorylated peptides that are co-enriched. And we have to get rid sort of these phosphorylated peptides because they might mask or they might hint the detection of the cross-linked species. This is one problem that we have, definitely. I know this is the main problem. So we have to introduce somewhere, as the previous speaker did, phosphatase treatment to remove, but only to remove the phosphate groups from the phosphorylated peptides, but keeping at least the RNA sort of partially phosphorylated so that we can enrich.
Yeah.
This
really,
is important. But
yeah.
When I think about this now, maybe we should do this afterwards, after the enrichment, then treat it with phosphatase and then spread again. Maybe this is worse, but this is in my opinion, the biggest bottleneck, or the biggest bottleneck, or the bottleneck when doing this in mammalian cells. But it is possible.
Yeah.
It's possible. We also cross-link now nuclei. We can cross-link cytosols, and of course, the ribosomes. The background, the same as you have in RNA modifications. The ribosomes, always the ribosomes, always DNA, the histones. These are the high abundant species that we have to remove as well.
Yeah, but this will be incredible if you get the data set. And of course, if not only mapping all the RNA binding surfaces, but if you can actually sequence the RNA attached to every surface on every protein, that would be even more incredible. We have just one quick question. This was Francisco Navarrete. He was saying, in your 70S ribosome, do you see evidence of what you might think are tRNAs cross-linked in the A site of the ribosome?
Yeah, actually we would like to, but we can not interpret this data, because we cannot distinguish whether the cross-link derived from, let's say, 20S or 16S, from the ribosomal RNA, or from a tRNA, But when we have this would be a very interesting experiment when we have a modified tRNA then and cross-link this.
I think he was thinking because the tRNAs are so modified, you might be able to get a unique signature that might tell you this is actually a tRNA. I can actually see the tRNA.
Yes, this could be possible, definitely.
All right, wonderful. And if questions pop up, if any of the audience members have questions, put them in the Q&A box, and Henning may be able to answer them over time. All right, now let's switch to Yinsheng Wang. Yinsheng is from University of California at Riverside, and he's going to talk to us about mass spectrometry for revealing the roles of RNA methylation in nucleotide repeat expansion disease. And we read this paper for Journal Club, so I'm going to be excited to hear this. All right, Yinsheng, thank you.
Yeah, all right. So
we can see your slides. You can go into full screen mode.
Yeah, let me show the laser pointer. Yeah. All right. Yeah. Thank you, Samie, for organizing this great symposium and for giving me the opportunity to speak. Yeah. I have enjoyed all the presentations this morning so far. Yeah. So as Samie shared, I'm going to share with you our recently published work together with some unpublished data about the mechanisms and the potential therapeutic implications. Yeah. So anyway, so I think many of you in the audience probably are familiar with the nucleotide repeat expansion diseases. Yeah. So actually, there are multiple neurological diseases. They arise from nucleotide repeat expansion. And one of the best known ones is really the CAG repeat expansion. So this one can happen in multiple genes, and that can have important implications in disease like Huntington's disease or multiple forms of spinal cerebellar ataxia. So one of the defining features for those diseases is that the repeat lengths, play a very crucial role. So for instance, for Huntington's disease, so most of us, will have this repeat. Actually, all of us have this repeat. But the length, can vary anywhere, say between 6 to 34 for healthy humans. Yeah. But for anyone with 40 or more repeats of CAG in the first exon of the Huntington gene, inevitably they will develop Huntington's disease in their lifetime. So the question is how, right? Why, you go from 34 being no disease, up to 40, then you have 100% disease penetration. So there have been multiple mechanisms proposed, about how the repeat expansion may contribute to neurodegeneration. So one mechanism states that, because the repeat, the DNA can be transcribed to mRNA, and some of those repeat RNA, they can be bound by cellular proteins. So now if the repeat is very, very long, so basically you sequester very important RNA binding proteins. So then you have a loss function of RNA binding proteins, right? And the second proposed mechanism is that this repeat RNA, they actually can be translated into proteins, right? So because the protein sequence is highly redundant, so therefore those proteins tend to not fold appropriately. So thereby giving rise to proteotoxic stress. So, but from our perspective, neither of these two mechanisms can explain fully, for example, for Huntington's disease, going from 34 being no disease up to 40 being 100% disease penetration. So we think that there must be additional mechanisms, that contributes to the disease etiology. I think all of this audience probably know that, the RNA now, is known to undergo reversible post-transcriptional modification, right? You have dedicated enzymes that can add, for example, a methyl group to various positions of the nucleic base or the deoxyribose or the ribose ring. And also there are specific enzymes that can take off these modifications. And then moreover, this modified nucleosides can be recognized by cellular proteins, right? So this has been shown to be very critical. You have seen this in previous talks already, in modulating the stability of tRNA and regulating translation efficiency, right? And furthermore, in mRNA, this can regulate the stability, translation efficiency, and alternative splicing of mRNA. So based on this prior work, we hypothesize that, maybe the nucleosides in the repeat expansion RNA, they can also be modified. And perhaps the modification frequency can be influenced by repeat length. So then, we decide to test this directly. So using LC-MS-MS. So this just show you, that with LC-MS-MS, we can unambiguously determine the M1A and the M6A, say in isolated RNA, because they can be chromatography resolved from each other very well. And also with the stable isotope standards, we can reliably determine the quantity or the frequencies of M1A and M6A, say in CAG repeat RNA. So then we test this directly by ectopically express the CAG repeat expansion RNA in cultured human cells with different repeat lengths. So with affinity chromatography, by using the CTG repeat DNA that's biotinate, so we can selectively enrich the CAG repeat RNA, from human cells. So then we can digest them down to single nucleosides, then we measure them by LC-MS-MS. So you can see that plot here is really the percentage of A that get methylated to M1A. So you can see that we observe a repeat length dependent increase in the modification frequency of M1A. Whereas for M6A, you did not see the repeat length dependent increase. So then we collaborate with multiple research groups, in different animal models. We examine how the repeat expansion may modulate the M1A frequency. You can see that the left part is a transgenic mouse model for human Huntington's disease. So in this case, it replaced one allele of the mouse HTT gene with a human counterpart, but either with or without the CAG repeat expansion. You can see that in this case, they use 140 repeats of CAG. So in the striatum tissue, the longer repeats clearly confers elevated level of M1A. But on the other hand, the cortex tissue, you did not see that. And I should emphasize that in Huntington's disease, the primary tissue that can impact is the striatum tissue. And the cortex only undergo neurodegeneration in the later stage of the disease. Yeah. So the second model is a Drosophila model for human SCAR3. So in this case, you have either 27 repeats of CAG that's considered normal, or 78 repeats of CAG that's considered pathogenic. That's in the human ataxin-3 gene to mimic SCAR3. So again, see that longer repeats give rise to high level of M1A. So the third model is the C. elegans model with the paneuronal expression of 19, 40, or 67 repeats of CAG. So you again observe a repeat length dependent increase in the M1A level in the repeat RNA. And for M6A, you did not see that. Yeah. So we did not see a repeat length dependent increase of M6A. So then we wondered why. Why the longer the repeat, can confer higher level M1A in the repeat RNA. So we reason that in applications data, perhaps the repeat RNA can somehow regulate either the demethylase for M1A or the methyltransferase for M1A. Right? So it's for the demethylation, for M1A, is primarily carried out by LQPG3 and to some degree also by LQPG1. And for M6A, is mainly demethylated by LQPG5 and FTO. And they do this, through a very similar oxidative demethylation mechanism. So basically oxidize the methyl group to generate hydroxymethyl functionality. So then they can undergo spontaneous loss of formaldehyde to regenerate the unmethylated A. So we examine which demethylase is involved in removal of M1A in the repeat RNA. You can see that only when we overexpress LQPG3, we see a pronounced drop in M1A in the CAG repeat RNA. LQPG1, FTO, LQPG5, do not have any impact. So this demonstrates that LQPG3 is a major demethylase for M1A in the CAG repeat RNA. So we also explored which methyltransferase is involved in M1A deposition. So we found that when we knock down TRMB61A, we see a pronounced drop in M1A in the CAG repeat RNA. We also checked other M1A methyltransferase for tRNA, and we did not see a pronounced change in M1A level for the CAG repeat RNA. So this really defines what's a methyltransferase and what's a demethylase for M1A in the CAG repeat RNA. So then we examine how the repeat RNA can modulate the expression of the demethylase. You can see that indeed we found that based on Western blot analysis, the expression of the CAG repeat RNA can give rise to pronounced drop in the LQPG3, the demethylase, right? And this effect is repeat length dependent. And similarly, we can observe the decrease in LQPG3 level in striatum tissue of the transgenic mouse of human Huntington's disease. And in cortex tissue, we did not see a pronounced change. So this mirrors what we observed for the M1A level seen in the two tissue types in the HD mouse model. So then you may wonder why, right? How the CAG repeat RNA can somehow suppress the demethylase. So actually many years ago, people found that the CAG repeat RNA, they can be processed by dicer machinery to produce 21-mer microRNA that's comprised of a CAG repeat. And this process is CAG length dependent, right? So you see here, with 70 repeats of CAG, you can see it. But with a shorter repeats of 30 of CAG repeats, you don't see that. And also it's dicer dependent, because if you remove dicer, this 21-mer microRNA can no longer be detectable, right? And a similar findings can be made, for the mouse model of Huntington's disease. And it's more so for striatum tissue than the cortex, right? And similarly, this microRNA can be seen in human brain tissue of Huntington's disease patients. And it's present also at a slightly higher level in the caudate, which is part of the striatum, than the frontal cortex. So also we checked the 3' UTR of LQPG3. It turns out that the 3' UTR, there's a short stretch of sequence, of LQPG3 that's highly complementary to the CAG repeat. So then, based on this, we reason that perhaps the 21-mer microRNA of the CAG repeat, they can hybridize with the 3' UTR of the demethylase LQPG3, thereby silence this gene. So we test this directly by transfecting cells with siRNA, either the SI control non-targeting sequence or the CAG repeat 21-mer small RNA, or the scrambled sequence ACG repeated by 7 times. You can see that only when we treat it with CAG repeat small interference RNA, we can see a pronounced decrease in LQPG3 protein level. So furthermore, we also generate a luciferase reporter assay. We found that, the 3' UTR is indeed responsive toward a synthetic CAG repeat RNA or toward a plasmid that allows for the expression of CAG repeat RNA. And this process depends on ARGANOT2, because when we knock down ARGANOT2, LQPG3, the luciferase reporter activity can be restored. So based on this, we basically made an interesting observation that, the longer the CAG repeat RNA, the higher the level of M1A right? So this mechanistically is because the CAG repeat RNA that can be processed by a dicer to generate a 21-mer CAG repeat microRNA. And this microRNA can be loaded to the RISC complex together with ARCB3 messenger RNA, thereby inducing the silencing of ARCB3. So this explains, why the longer the repeat, the lower the level of ARCB3 and the high level M1A. That's because with the repeat RNA, can be processed to microRNA, the silenced, the demethylates ARCB3. So with that, so then we wondered how this modification in RNA really can modulate RNA-protein interactions. So we think that perhaps, the M1A modification, they can change the RNA-protein interaction homeostasis. So we decided to examine how the M1A are recognized by cellular proteins. Yeah. So we developed a quantitative proteomic method to screen for proteins that can bind specifically to M1A. So this is a biotinate RNA probe, derived from the human telomere RNA sequence with a defined M1A site, with a single M1A or without any modification. So through this analysis, we were able to uncover a bunch of candidate M1A binding proteins. And the top hit, we identified in HeLa cells, it turns out to be a protein called a TAR-DBP, also known as TDP43. So this is mass spec data. You can see that in the forward SILAC screening, where the M1A containing probe is incubated with heavy lysate, and the unmodified RNA is incubated with light lysate. You see selective binding toward M1A containing RNA. So then we swap the incubation. So in the reverse SILAC incubation, so we again see the TDP43 binds more strongly to M1A containing RNA over unmodified RNA. I mean, proteomic method is powerful, but sometimes we are not sure, whether the protein binds to RNA is direct or through protein-protein interactions. So we have to validate, whether this interaction is direct. So to test that, we purified recombinant TDP43. So then with the gel shift assay, and we found that indeed TDP43 can bind selectively to M1A over the A or M6A containing substrate. Yeah. And similarly with the CAG repeat RNA, so they also the TDP43 prefers the M1A containing CAG repeat RNA over the unmodified RNA. So TDP43 is a very interesting protein in the neurodegeneration field because there is a phenomenon known as the TDP43 proteinopathy. So this has been found, in many neurological diseases, including Huntington's disease, FTLD, and ALS. So the key phenomenon is that, the protein displays cytoplasmic distribution and forming inclusion bodies. Because TDP43 normally is present in the nuclei, but in those diseases, they become cytoplasmic redistributed and form inclusion bodies. And also they can undergo C-terminal truncation, phosphorylation, ubiquitination. So with this, we come up with a sort of hypothesis. Maybe, M1A, we reason that CAG repeat RNA, because they're heavily decorated with M1A, right? So they may serve as a seed, for TDP43 aggregation. Because TDP43 is a protein, that's known to susceptible to aggregation. So now you have a CAG repeat RNA, they are decorated with M1A here and there. So this basically increases the local concentration of TDP43 surrounding the CAG repeat RNA, so thereby promoting its aggregation. So then eventually leading to TDP43 proteinopathy. So we test this directly. So this you can see that is in U2S cells, with the EGFR tagged TDP43. Without the CAG repeat expansion RNA, the protein is primarily in the nuclei. So now as soon as we ectopically co-express the CAG repeat RNA, so you see the cytoplasmic redistribution of the TDP43 protein. And the longer the repeat, the larger the foci size you can see for the TDP43 protein. And this can be rescued if we overexpress human ARCB3. So if we overexpress the demethylase, the cytoplasmic distribution becomes diminished. And also we found that by using RNA fish and together with immunofluorescence microscopy, we found that the TDP43 is co-localized with the CAG repeat RNA. And furthermore, we show that this co-localization actually occurs in stress granules because they also co-localize with the G3PP1. And then next we examine whether this can also give rise to TDP43 truncation. We found that indeed in the detergent insoluble fraction, we can see that with overexpression of CAG repeat RNA, we can detect the truncated TDP43. And the longer the repeat, the higher the degree of this truncation. And similar findings can be made for the stratum tissue of a transgenic mouse model of human Huntington disease, and it cannot be seen for the cortex. So this parallels what we observe for the M1A level in the CAG repeat RNA isolated from these tissue types. And next we examine how this interaction between CAG repeat RNA and the TDP43 can affect the biophysical properties of TDP43. So we conduct fluorescence recovery of the photobleaching experiment. You can see that when you express the shorter repeats of CAG, 22 repeats, the protein is still dynamic. So after photobleaching, the fluorescence intensity can recover in a reasonably short period of time. However, with the long repeats of CAG, 38 repeats of CAG, you can see that recovery is much slower, right? And also the degree of recovery is much poorer. But in this condition, when we overexpress LKP3, the demethylase, so you can see that the protein becomes dynamic again, right? So this really basically demonstrates that is M1A in the CAG repeat RNA that drives the static protein behavior of TDP43. And also we test the phase separation behavior of the TDP43 protein. Right? You can see that this with 22 repeats of CAG. So after you treat the cells with 1,6-hexane diol, which can disrupt physiologic phase separated droplets, because it can diminish the hydrophobic interactions. You can see that the droplets can be dissolved. But with 38 repeats of CAG, so now the droplets can no longer be dissolved, right? So in other words, this long repeats of CAG repeat RNA converts the protein from a physiologic phase separated condensate to gel-like aggregates. So we also examining whether this contributes to neurodegeneration. So in the C. elegans model, with the expression of 6-7 repeats of CAG, you can start to see the gaps in the neural cord. And these gaps, actually can be resealed if we overexpress human LKP3, right? And also it can be partially resealed, if we can downregulate the worm ortholog of human TRMD61A by using RNAi. And the control RNAi cannot do that. And likewise, in Drosophila model of SCAR3, we found that when we ectopically express human wild-type LKP3, actually we have elongated lifespan of the fly, where the catalytic dead enzyme cannot do that, right? And M1A measurement show that wild-type human LKP3 can reduce M1A level. The catalytic dead enzyme cannot do that, and M6A level is not affected. And with that, we think that perhaps this TRMD61A may serve as an important therapeutic target, for HD, right? So we collaborated with Professor Jianpeng Cai at the University of South Florida. We developed a series of cyclic peptide inhibitors for TRMD61A, and we found that indeed, in the cultural cell line model at least, the treatment of the cyclic peptides, they can diminish TDP43 truncation. So with that, I want to summarize what I have shown you. So basically, we found that the A in the CAG repeat RNA can be methylated to M1A and M6A, and the frequency of M1A increases with repeat length. And mechanistically, this is because the CAG repeat RNA that can be processed to microRNA that can silence the demethylase. And also, we found that this M1A drives the change of the TDP43, from the physiologic phase separated condensate into gel-like aggregates. So basically, the take-home message is that an RNA modification play a very crucial role in CAG repeat expansion diseases. Yeah. So I want to thank my co-workers. Yinsheng Sun is a postdoc who leads this effort and is supported by another postdoc, Hui Dai. And the recent work on cyclic peptides done by Hongjun Zhang, postdoc, current postdoc, and all the collaborators for the different animal models and for generating cyclic peptides. And then the funding from NIEHS. Yeah, with that, I want to thank you for your attention. Happy to take any questions. Yeah.
All right. What a great and very interesting and medically relevant talk. I've got two questions for you.
Yeah.
As we wait for people to ask questions, although we are running a little bit late.
Yeah.
The enzyme that makes M1A, at least in tRNA, operates in a very specific structural context and sequence context. Do you think this is the same enzyme that makes M1A in the CAG repeat?
Yeah, we did some in vitro experiment with purified TRMD661 complex. It can methylate CAG repeat RNA to M1A, but not as efficient as the tRNA. And
So-
then with RNAi experiment, when we knock down 61A, but not other mRNA methyltransferase like 10C or 61B, we can see the pronounced decrease of mRNA in the CAG repeat RNA. I mean, there's some structure analogy, because in tRNA, the mRNA is present in this T loop, right?
Yeah.
And the A in the CAG repeat RNA can also loop up,
Yeah,
Yeah, but the loop is much smaller, of course, in this case. Yeah.
Yeah, very interesting. Now,
Thank you.
TDP-43 aggregates in not just Huntington's disease, it's actually
Absolutely.
much more prominent aggregation in ALS, in frontotemporal dementia. Do you think that there is an M1A RNA that's aggregating there, or that the RNA in TDP-43 aggregates has M1A there, which is helping to facilitate those TDP-43 aggregates in other diseases?
Most likely, yeah. I did not show you the data. We collaborate with Fei-Hong Chen at a Cleveland clinic. Yeah, we look at Alzheimer's disease.
Wow.
We look at patient tissues. Yeah, we see elevated level M1A as well, and that's correlated with TDP-43 pathology.
Got it.
Also, I did not show you the data today here. Even without CAG repeat expansion RNA, if we downregulate ALK3 by using the CAG repeat microRNA, we can also see lower degree of TDP-43 pathology. Just CAG repeat expansion RNA exacerbates the process. Yeah.
Wow. That would be amazing if by blocking the M1A formation, you could really help these people with TDP-43 diseases. I have other questions, but we really have to end. People can enter questions in the chat box, and Yinsheng may be able to address them in 1 hour, or slightly less than 1 hour, but 1 hour. We're going to be back on with the second session. I hope you guys can join us. It's really a very interesting set of speakers for the next set. I apologize for the people in China and Japan. It's very, very late, but hopefully people can join us in 1 hour, and we'll see you soon.
Yeah, thank you. See you soon. Yeah.
Bye.